Hello everyone. In the last class, we discussed about the Mendel's law, and today we will be discussing about law of independent assortment and the non-Mendelian law. So let's get started. So before starting law of independent assortment, we shall discuss about the dihybrid cross. In the previous class, we discussed about monohybrid cross. So repeating what I said in the previous class, monohybrid cross is the cross which is performed taking only one character in consideration. For example, seed shape. The dominant and recessive seed shapes are made to cross with each other. This is known as a monohybrid cross. Here we are talking about a dihybrid cross, which means that when two characters are taken in consideration, it is known as a dihybrid cross. Here we have taken the characters seed shape and seed color. The dominant seed shape is round and the dominant seed color is yellow. So you can see one of our parent is capital Y, capital Y, capital R, that is round yellow dominant parent, and the other parent is small y, small r, that is the wrinkled green recessive parent. So when they are crossed, we get our F1 generation as the heterozygous as the heterozygous progeny, which is capital Y, capital R, small r, that is which displays a color, expresses a color of yellow round. When the F1 is made to cross, it forms for the F2 generation. The F2 cross has been shown in the Punnett square. So you can see that there are total 9 round yellow and 3 round green, 3 wrinkled yellow and 1 wrinkled green progenies, which makes the phenotypic ratio as 9:3:3:1. Round yellow 9, round green 3, wrinkled yellow 3, wrinkled green 1. That is 9:3:3:1. We do not take genotypic ratio for at your level for now. So moving forward to our law of independent assortment. Law of independent assortment is all about that the factors for different pairs of traits assort independently of one another. This means that during meiosis, when the gene pairs are separating to form gametes, one pair of gene does not affect the other pair of gene while getting separated. It is their individuality, on their individual basis they get separated. This is what has been explained in law of independent assortment. In modern terms, we can say that pairs of alleles for genes on different chromosomes segregate independently in the formation of gametes. That is, for example, that we have a heterozygous yellow colored seed with us. Heterozygous means one of the allele is capital Y and one of the allele is small y. So when capital Y, small y gene pairs segregate in meiosis, then capital Y and small y do not interfere in each other's process and they segregate independently without affecting the other one's function or the functionality. Moving forward, now we are going to start with the non-Mendelian laws. At present, we will be discussing about two non-Mendelian laws, incomplete dominance and codominance. So what is incomplete dominance? As you can clearly see in the picture, incomplete dominance is most of the time observed in dog flower, also known as snapdragon or antirrhenium species. The flower color is affected in this case. In such case, the genotypic ratio remains the same, but the phenotypic ratio is affected. When we are talking about the monohybrid cross, the normal genotypic ratio for monohybrid cross remains the same, which is 1:2:1, whereas the phenotypic ratio, which is supposed to be 3:1, is affected in this case. As you can clearly see from the picture, that in the F1 generation, which is supposed to resemble either of the parents, showcases a trait in between the two parents. One of the parent is in red color, which is the dominant trait, capital R, and other parent is in white color, which is a recessive trait, which generally represented by small r, or we can also represent it by WW. So here you can see the F1 progeny found is in pink color, which should have been red, because generally, according to the Mendel's law, the F1 generation depicts the dominant character of the parents, but here it has taken up the character in between both the parents. When we cross this F1 generation, that is when we cross RW, then we get in the F2 generation, the phenotypic ratio which is received is 1:2:1. That is 1 red, 2 pink and 1 white flower. Whereas the genotypic ratio, which is for Mendelian laws 1:2:1, remains the same for incomplete dominance as well. Moving forward, here are some tricks to determine the number of genotypes and phenotypes expressed by the progeny. The formulas are phenotype 2 raised to the power n, and for genotype 3 raised to the power n, where n is the number of different genes. For example, here we have taken the example of capital A, small a, capital B, small b. Whenever we have a gene pair in heterozygous form like capital A, small a, we consider it as 1. It is adding a value of 1. So here we have 2 heterozygous pairs. So our n, that is the number of different genes, will be 2. So the phenotype received here will be 2 raised to the power 2, which is total of 4 phenotypes, and the genotype will be 3 raised to the power 2, which is total of 9 genotypes. You can refer to our notes for better clarification, and you can also post your doubts in the comment section. Next up, we have the Codominance Law, which is observed in the ABO blood grouping system. ABO blood grouping is controlled by I gene, which has 3 alleles, IA, IB and small I. Therefore, it is also an example of multiple allele. The surface of RBC, as you can see in the picture, has sugar receptors which are controlled by I gene. These blue color are present in the antigen A, these triangles or spike kind of things present in antigen B, these are the sugar receptors which are controlled by I gene. So, when IA allele and IB allele produce slightly different kinds of sugar from each other, whereas small I allele produce no sugar. You can also see this from the picture. For antigen A, we have blue color spike kind of proteins, spike kind of sugar receptors coming out present at the surface of RBC, whereas for antigen B, that is for blood group B, we have spike-like proteins, spike-like sugar receptors on the surface of RBC. And when we talk about the last case, that is the type O, there is no sugar receptor present on the surface of the RBC. This is the small I allele. This kind of sugar receptor, what kind of sugar receptor is to be present on the RBC, is controlled by I gene. IA, that is the antigen A, and antigen B, both are of dominant type. So when they are present together, that is in the case of AB blood group, how will we decide, or how is it decided that which allele will depict itself or express itself? Because both antigen A and antigen B, that is both the alleles A and B, are dominant in nature. So in this case they show codominance, that is both of them, that is both allele A and allele B, express themselves at the same time. When two things express or function at the same time, it is known as codominance, as the dominant level is being coordinated. So we call it as codominance. This is the case of codominance. So when we have a case of capital IA small I or capital IA, the blood group we get is A blood group. When we have a case of capital IB small I, capital IB, we have the B blood group. And when small I is present, we have O blood group. Why we have only one gene pair for O blood group? Because small I is a recessive allele. So only in homozygous condition it expresses itself. And when allele capital IA, capital IB, both are present together, the blood group attained is AB. Once again I repeat, both alleles IA and IB express themselves in the case of AB blood group, and therefore it is known as codominance. With this, we have come to the end of this video. In next video, we shall be discussing about chromosomal theory of inheritance. Till then, stay tuned and keep learning.